Once upon a time, there was a professor known as superhero Dr. Chu.
Her superpower was applying bioenvironmental technology to real-world problems.
She had a great idea. Let's create a class that will teach students about wastewater treatment plants along with nitrification and denitrification.
She found 6 enthusiastic researchers and 2 knowledgeable graduate student TAs to help.
Together, we formed a plan. Why not use anammox and nitrification to achieve a high nitrogen removal rate from wastewater? We immediately started trying to animate our anammox. John grabbed a sample from a wastewater treatment plant in Hearne, Texas. We fed them every week for 4 weeks.
We fed them bicarbonate for a carbon source, nitrite as an electron acceptor, and ammonia as an electron donor. Both the nitrite and ammonia are used for the nitrogen source.
In the meantime, while our anammox were growing, Ted and Lee taught us analytical methods in the lab.
We did some literature review to apply others' research to our own. We became experts at pipetting and weighing to be precise in our solutions. We made synthetic waste to use in our anammox experiment. We did some DNA extraction, PCR, and electrophoresis to visualize the anammox.
Use a spectrophotometer and hatch kit to create a standard curve to measure any nutrients.
After about 5 weeks, we extracted DNA from our sample. We amplified the DNA through PCR, but when we viewed it under electrophoresis, there was none.
We were lost, but superhero Dr. Chu came to help.
Dr. Chu found an institution in Virginia that had already grown anammox, so he had them send us a sample.
So after Thanksgiving, we enriched the starter sludge, extracted DNA, ran PCR, and this time under electrophoresis, we could confirm that we had anammox and AOB.
In the future, we will combine nitrification and the anammox reactions in a sequential batch reactor. Because the anammox don't like the oxygen-filled environment needed for nitrification, Lazar will encapsulate the anammox into gel beads. Anammox are also very slow growers, so these beads will give them their own anoxic home, complete with the long retention time. The end result is very happy anammox.
Here is our proposed reactor. We are going to add synthetic wastewater, the AOB as the nitrification agent, the encapsulated anammox. They will react together and give us nitrogen gas. Yay!
The anticipated benefits of this reactor is low energy requirements, lower oxygen demand, less sludge creation, and a high rate of nitrogen removal. This proposed process we call SNARE, single reactor nitrification and ammonia removal using encapsulated anammox.